Hello, good evening everybody. So today we will be summarizing whatever we have gone through in all of these weeks, what we have understood in immunology, okay, and then we'll take few questions also from all of those parts, okay. So this is immunology course. So we discussed about how what is this immunology. It is a study of immune system and defense mechanisms, okay. And as a defense mechanism, what are these functions of the immune system? So in our body, the immune system, it has a function in defense against the foreign pathogens, okay. And it function in the removal of the damaged cells and also in the surveillance against tumors. So it's basically a balance between protection and tolerance, okay. So we'll have gone through in this entire immunology course what is immunology, okay, and what are the branches of immunology: innate, adaptive immunity, what are the cells and organs that are involved in this particular immune system, and what are the antigens, what is antibodies, what is their structure, what are their functions, and then how the antigen is being processed and being presented to the immune cells, and what is the effector mechanism to kill those particular foreign pathogen and infected human cells, okay. So all these are the overview of the immune system. So we'll go one by one, okay. So immune system, we know it is a defense mechanism of our body where it fights against the foreign pathogen or the defective cells and try to clear them out in order to protect our body, okay. So this particular phenomena is called immunity. The immunity could be of two types: innate immunity as well as adaptive immunity, okay. So the innate immunity is basically the first line of defense for our human body. So innate immunity is a non-specific. That means as being a first line defense, whatever infection or pathogen I get infected with, okay, any kind, whether it is a virus, it is a bacteria, it is a fungus, or any other thing, okay, any other parasite. So this is the first defense which stops them. So this is innate, okay, and thus it is non-specific because it is not very efficient, but it fights with all. So it is a non-specific immunity. It is immediate, okay, for a quick action, as being a first line defense mechanism. And also it does not have any memory, okay. Like if the same pathogen will attack again, the first line of defense, the innate immunity, does not have any memory against it, okay. So these are the classification, but these are the first line of defense which helps protecting us in immediately whenever any infection happens or occurs. Then if that particular pathogen cannot be taken care by the innate immunity of our body, then comes the adaptive immunity, okay, which is very specific, okay, and it has a little delayed response. It is not that immediate because as it is specific, it goes through certain set of cells, okay, and the antigen needs to be processed in order to be presented to the immune cells, and that is why it is delayed. And when the action or reactions of immune system takes place, those cells which are generated in this immune defense mechanism, they also generate a memory against that particular foreign pathogen, okay. So whenever there is a next infection of the same pathogen, our adaptive immune system is ready and react more efficiently to that particular pathogen because of the memory, okay. So in the second attack, it is not delayed, it is an immediate action kind of thing, okay. So for example, here you can see this is the primary immune response and this is the secondary immune response. So if in the adaptive immunity we'll talk about the antibody concentration, so it is taking, as you can see from this graph, it is taking a little time. Slowly it is increasing, and antibodies are generated in the primary immune response in the initial exposure to antigen. Then it gradually lowers down. Whenever there is a second exposure to this antigen, now you can see compared to this one, this is rather quick. Okay, this is quicker, and now you can see the antibody concentration is also very high in the secondary immune response. Okay, so the response is faster, response is larger. Okay, so it is also mentioned here that once a foreign organism has been recognized, the immune system recruits a variety of cells and molecules to mount an approximate response called an effector response to eliminate or neutralize the organism. Later exposure to the same foreign organism may induce a memory response only in case of adaptive immunity. Okay, a more rapid and heightened immune reaction, not in case of the innate immunity, because innate immunity does not have any memory. Okay, so in case of adaptive immunity, due to the memory response, it is having a heightened immune reaction. Okay, so here you can see, so this is, you can see basically the first line of defense. Okay, here the physiological barriers also you can say. Skin. Okay, whenever, so our skin is also a physiological barrier that protects us from any kind of foreign pathogen or anything. Okay, you cannot just, it cannot just enter our skin. Okay, so skin is works as a kind of protection or physiological barrier against the pathogens. And then there is a mucous membrane. Okay, so a pathogen tries to enter our via our mouth cavity, oral cavity. Then also there is a mucous membranes which also um, kind of stops them. Then there is a cilia through our GI tract. Then the body temperature, if the body temperature is high, the foreign pathogen will not survive. The pH of those also provide the physical barriers. Okay, so that the pathogens will not be harming us. And there is innate immunity. In the innate immunity, it is kind of non-specific. The neutrophils, okay, WBC is basically the neutrophils are involved. Okay, basophils are involved, eosinophil, macrophages, mast cell, dendritic cell, natural killer cell, all these kind of cells are involved in the innate immunity, which shows immediate reaction. Okay, in form of phagocytosis. Okay, they internalize the foreign pathogen, lyse them, phagolysosome complex they make, they lyse them. Okay, so those are the innate immunity. It is immediate and non-specific. Okay, then comes the adaptive immunity, which is very specific. It involves the T or B lymphocytes. Okay, and here also you can see this is the response, the same graph, initial exposure and the second exposure. So here you can see in the primary immune response, so in the primary immune response, you can see here initial exposure of antigen A. Okay, this blue graph, it shows the primary response delayed. Okay, again when there is a secondary exposure of this antigen A, now you can say there is a heightened response, more antibody concentration against antigen A. At this time point, when there is a exposure to another antigen, antigen B, as against this antigen the body does not have any memory, then it is again a delayed response compared to the A, for which the body has the memory. So it is a heightened response and this is a delayed response, because, and it is also not as heightened as of the second exposure of A. So this is the difference. Okay, so yeah, innate immunity, which is the first line, it is non-specific, immediate defense, minutes to hours. So if we talk about the components, the physical barrier, skin, mucosa, cilia, secretions, lysozymes, defensins. Okay, the cells that are involved, those are macrophages, neutrophils, dendritic cells, natural killer cells. There are soluble molecules, for example complement proteins, interferons, acute phase proteins. There are some pattern recognition receptors also present, which recognize these PAMs or DAMs, or they are having, for example, toll-like receptors. Okay, and so these are pattern associated some proteins. Okay, so those are being recognized by this toll-like receptor, which are part of the innate immunity. So the key feature of this innate immunity, it has no memory, and it has same response on repeated exposure, okay, because there is no memory. Second is the adaptive immunity, which is second line of defense, okay. So this adaptive immunity, it is specific, it has memory, so it takes certain days to be activated, okay. And this specific or adaptive immunity, it has two arms. So it is divided into humoral immunity and the cell mediated immunity, okay. So in the humoral immunity, you have the B lymphocytes involvement, okay, and in the cell mediated immunity, you have the T cell lymphocytes involvement. So basically, when you talk about the cell mediated, so the cell is itself involved, okay, and in killing, on clearing out the pathogen. But in the humoral immunity, the B cell, they make the plasma cells, and then plasma cells release the antibody in the what you can say, the fluid, in the body fluid, and those antibodies try to um, kill the pathogens, interact and kill the pathogens. So that is in humoral immunity, okay. But when the cell is itself involved, that happens in case of T cell mediated immunity, okay. So the feature of this kind of immunity, it is specific, it has memory, and it can also discriminate between self and non-self, okay. Okay. So yeah, so how these cells are arised, these immune cells, okay. So in the stem cell, okay, in the bone marrow, it generates two kinds of progenitor cell, lymphoid progenitor and myeloid progenitor cells. So the myeloid ones, they generates all these cells of the innate immunity: neutrophils, eosinophils, basophils, mast cells, monocytes, dendritic cells, and macrophages, okay. So these dendritic cells, okay, these are the professional antigen presenting cells that display antigens to activate the naive T lymphocytes, okay. So these are the cells which present the antigen to the T cells, and T cells are part of the lymphoid progenitor cells or part of the adaptive immunity. So all these cells which are part of the, what you can say, the innate immunity, only one of those cells, which is dendritic cells, which present the antigen to the T cell, which is part of the adaptive immunity. It is a kind of a link between the innate and adaptive immunity, okay. So these neutrophils, these are the phagocytic cells that rapidly migrate to the site of infection, okay, to ingest invading microorganism. They release toxins to combat the bacteria and fungi and recruit other immune cells to the site of infection, okay. There are eosinophils, which are also phagocytic cells, okay. These basophils, non-phagocytic cells responsible for defense against parasite and have the FC receptor for IgE, okay. Mast cells, these are the granulated tissue cells that degranulate in response to the tissue injury and initiate the inflammatory reaction through the vasoreactive properties of histamine. Monocytes, these migrate to the site of inflammation and mature into tissue macrophages and dendritic cells, okay. So macrophages, these are large phagocytic cells that ingest extracellular debris and invaders and consequently uh, stimulating other immune cells, okay. So these dendritic cell is a professional antigen presenting cells, okay. Then there is a lymphoid progenitor which produces T cell progenitor and B cells. So it basically produces the T and B lymphocytes, okay. So the T lymphocytes, it can be divided into T helper and cytotoxic T cells, okay. So cytotoxic T cells, this kill virus infected and tumor infected cells, and these helper cells, so what they do, they mediate the inflammatory reaction, immunity to intracellular microbes, or help the B lymphocyte to produce the antibody. So these are basically the helper cells, and this is the killing cell, the cytotoxic T cells. Then there is the B progenitor cells. B progenitor, upon activation, it makes memory cells and plasma cells, Okay, so plasma cells, they manufacture large volumes of antibodies. Memory cells are the long-lived lymphocytes capable of responding to particular antigen on its reintroduction long after exposure that prompted its production. Okay, and then there is this natural killer cell. These are the type of killer lymphocytes related to the CTL but lack the antigen receptor. Okay, like this one, cytotoxic cell, but it lacks the antigen receptors. Okay, so this is the basic summary. Okay, so mast cell, their function: allergy, inflammation, basophils, eosinophils. They're involved in defense against the parasite. Okay, natural killer cell, they can also kill the virus infected and tumor infected cells. That is same being done by the cytotoxic T cells as well, but there they have the antigen receptors, but the NK cells does not have the antigen receptor. So dendritic cells, they uh, help or work as antigen presenting cell. They are called as professional antigen presenting cells, and they are the bridge between innate and adaptive immunity. Neutrophils, they function in phagocytosis or inflammation, and they are the first responders to any infection, and they are very abundant in blood. Macrophages, they are their function is the phagocytosis, and they also help in the antigen presentation. Okay, they are tissue resident and long lived. Okay, and as part of the adaptive immunity, there are B cells and T cells and memory cells, you can say. So B cells, they make antibodies, and memory B cells, okay, the T cells, they mediate cell mediated immunity. Okay, they have CD4 positive T helper cells, then CD8 positive cytotoxic T cells. Then there are memory cells which have rapid strong response to the re-exposure. It is there in both B and T memory cells. So if we talk about the components of the immune system, so cells of the immune system, so innate, adaptive. So in the innate, you have these macrophages, which function is phagocytosis, antigen presentation. Neutrophils are the first responders, dendritic cells are the bridges between innate and adaptive. NK cells, they kill virus infected, tumor infected cells. Adaptive immunity, you have the B lymphocyte that produce the antibodies. Humoral immunity, and in the T lymphocyte, that is the cell mediated immunity, and they have two types: helper T cells and cytotoxic T cells, and they have a particular mark. So all helper T cells are having CD4 in their cell surface, so that is why they are called as CD4 positive cells. All the cytotoxic T cells, they have only this CD8 specifically present on the cytotoxic T cells as a marker, so these are also called CD8 positive cells. Okay, so CD4 positive helper T cells, they activate the B cells and macrophages. CD8 positive cytotoxic T cells, they kill the infected cells. Okay, and there are these other important cells, for example mast cell, eosinophils and basophils. And when we talk about the organs of the immune system, so these are the cells. Organs, so there are two kinds of organs: primary lymphoid organs and secondary lymphoid organs. So primary lymphoid organs are the bone marrow and the thymus, where these immune cells get mature, okay, develop, originate and develop and being matured. And the secondary lymphoid organs, where these immune cells are being activated. Okay, so those are the lymph nodes, spleen, mucosa associated lymphoid tissue, and etc. So basically the lymph node, spleen, all these are secondary lymphoid organs, where these these immune cells gets activated by facing the pathogen. So there they get to face the pathogen. Okay, so yeah, so in the thymus, you have the T cell development going on. In the lymph node, the antigen presentation takes place. In the bone marrow, the hematopoiesis and the B cell maturation takes place. Okay, so both these T cells and B cells, they get originated from bone marrow. B cells, they also being developed in the bone marrow, but the T cells, they go to the thymus for the development. Okay, so origin wise, both B lymphocytes as well as T lymphocytes, that originates in the bone marrow, but T lymphocytes go to thymus for the development rather than the B lymphocyte, which get matured and developed in the bone marrow only. Okay, So the primary lymphoid organs, so these are the sites where lymphocytes are generated and being mature. So bone marrow, so bone marrow is present in the central cavity of the bone, okay, the large bones, basically sternum, pelvis, femur. So these are the site of hematopoiesis, where production of the all blood cells, including the human immune cells as well. So here the B cell development and maturation occurs in mammals, and they produces common lymphoid and myeloid progenitors. They contain the stromal cells that provide growth factors and signals. Okay. In the thymus, it is a location, upper anterior mediastinum behind the sternum. Its function is the site of T cell maturation. Immature T cells, which are called as thymocytes from bone marrow, migrate here and undergo positive and negative selection. Okay, so what is positive and negative selection? So positive selection is, it ensures that T cell recognize the self MHC. Okay, so these thymocytes start developing. Okay, and then these thymocytes, or the naive T cell, uh, in the first selection process, they are being tested whether they can recognize the MHC molecules. So what are the MHC molecules? These are the major histocompatibility complex. So these are the molecules or present on the cell surface and help in presentation of the antigen. Okay, so if an antigen presentation cell is there on, it is present on the MHC is present on the surface of the antigen presenting cells, and the antigen is being processed from the antigen presenting cells and then goes to the MHC, and the MHC presents those antigens to the immune cells, which are T lymphocytes. Okay, so in order to be activated or to be selected to fight against the foreign pathogen, the thymocyte needs to learn whether it can recognize the antigen being presented by the MHC of self from our own body. Okay, so that is the first selection. So if it is recognizing antigen from the self MHC, that is the positive selection, the cells are selected. Okay, that is positive selection. Then there comes the negative selection. So it eliminates strongly self-reactive T cells. So the self MHC, it is presenting the antigen to the T lymphocytes. Okay, so the T cells, they undergo positive and negative selection inside the thymus, where they get mature. Okay, so positive selection is whether these T lymphocytes can recognize the antigen being presented by its own MHC molecule. Okay. So I'll just quickly tell one more time what I have told. So we have been discussing about what are the cells of the immune system, what are the organs of the immune system. Okay, so yeah, so basically, we have talked up till now as a summary class. We have talked about innate and adaptive immunity. Okay, we have talked about how this adaptive immunity is very specific. Okay, and the innate immunity, which is comes before the adaptive immunity, the pathogen has to cross it one by one. Okay, so the pathogen has to cross first the physical and physiological barriers, which include skin, mucous membrane, cilia, body temperature, pH. If the pathogen is able to pass that as well, okay, majority of the pathogen cannot pass that. If some of the portion of the pathogen which can even cross that physiological or physical barrier, they'll first interact the innate immunity. Okay, so in the innate immunity cells, so innate immunity cells contains this neutrophils, basophils, eosinophils, macrophages, mast cells, dendritic cells, and the natural killer cells. So all these cells have very immediate reaction to the foreign pathogen. Okay, and they are not specific in nature. Okay, be it virus, bacteria, fungus, parasite, any foreign pathogen which is not self. Okay, these innate immunity cells will respond against them, and that is a very quick immediate response. Okay, and try to kill them by phagocytosis or releasing some histamines or any chemicals and any cytokines any granzymes, granules, okay, they try to kill them. Okay, so that is the role of the innate immunity. Okay, and as it is very immediate, it is very non-specific, that's why they do not have any memory also. Okay, so if the same pathogen will act on, come and infect again, these innate immunity cells, they do not have any memory, but their action is immediate but not as effective as of adaptive immunity. So if the foreign pathogen is now able to cross the innate immunity barrier as well, it goes to the adaptive immunity. It will be taken care by the adaptive immunity. So in the adaptive immunity, we have T lymphocytes and B lymphocytes. They are very specific. They have the immune response by the adaptive immunity is kind of a little delayed compared to the innate immunity, because the antigen needs to be processed first from in order to be presented to the specific cells types and in order to be killed by those cells of the adaptive immune system. And these adaptive immune system cells, they also do possess a memory against the pathogen. So here you can, with this graph you can see the initial exposure to the antigen A, it is having a kind of delayed response. You can say few days are being taken, then the response starts, and this is the maximum response antibody concentration that is being produced in the primary immune response. Then again, when the same antigen is infecting, this blue graph you follow, again the antigen A is infecting our body. Now in the first response, in the adaptive immunity has made some memory cell against it, so now it recognizes this antigen quickly and give a quicker and a heightened response compared to the primary response. Okay, at the same time, if another pathogen which is infecting the body for the first time, as the adaptive immune system shows a fast response for antigen A, for antigen B which is infecting for the first time, it will again show a slow response because it has no memory, no prior memory, and this response is not as heightened as of the against the antigen A. Okay, so that is the difference. So yeah, so these are the progenitors for the B lymphocytes, B lymphocyte and T lymphocyte. The lymphoid progenitor, the myeloid progenitor, will produces all the cells which are involved in the innate immunity. It produces the dendritic cells which are called as professional antigen presenting cells. They present the antigen to the T lymphocytes which are part of the adaptive immunity. So that is why the dendritic cells are being called as the bridge between innate and adaptive immunity. Okay, so now we were talking about the components. So cells we have talked, the organs, so there are lymph nodes, bone marrow, thymus. So they are divided in primary lymphoid organs. So primary lymphoid organ is bone marrow, so bone marrow and thymus. So bone marrow, where the, so both T lymphocytes and the B lymphocytes are being originated in the bone marrow, but T lymphocytes go to the thymus for its development and maturation. However, the B lymphocyte gets matured in the bone marrow only. Okay, so in the thymus, when the T cell gets mature, it undergo positive and negative selection. Part of positive selection, that means the all those T cells or the naive T cells will be selected which can recognize its own body's MHC. Okay, so if my body's MHC molecule will present the antigen to the T cell, then only it should react to that. Okay, so that is the first selection. So positive selection, whatever T cells are recognizing the self MHC molecule or the antigen being presented to this by the self MHC molecule, they will be selected. So that is a positive selection. That is one check. Another one will be if the naive T cell is reacting very strongly to the self antigen, then that is not a good thing. Okay, in that response it will kill the self cells. Okay, so that is a negative selection. So whatever T lymphocyte which is having a very strong reactivity to the self proteins or self antigens, that cells will be eliminated. Okay, so after that only 5% of the T cells survive and enter the circulation as mature naive T cells. Okay, so age related evolution, so thymus shrinks and becomes less active after puberty, but peripheral T cell pool is maintained. Okay, so secondary lymphoid organs is the organs where the these immune cells, B lymphocyte and T lymphocyte, gets activated or face the antigen. Okay, so that includes the lymph node and the spleen and also other organs, for example malt and galt. Okay, so these are the sites where mature lymphocytes are activated by encountering antigens. So lymph nodes, so these are the encapsulated bean shaped structure along lymphatic vessels. The function of the lymph nodes, they filter the lymph for the antigens. They are the site for antigen presentation by dendritic cells to naive T and B cells. Okay, so you know dendritic cells are the part of the innate immunity, but as they are presenting the antigen to the T cells and B cells, they serve as a bridge between the innate and adaptive immunity. Okay, their function is also the lymphocyte trafficking. That means the T and B cell enter via blood, survey for antigen, exit via efferent lymphatics. Okay, they contains distinct zones. So they have cortex where the B cells are activated, germinal centers, in the germinal center specifically, paracortex where T cell resides, medulla for the plasma cells. Okay, then there is spleen, which is located upper the left abdominal quadrant. It function for the filtering of blood but not the lymph. It removes aged or damaged RBCs. The immune response to the blood-borne antigen, and it contains white pulp and red pulp. So white pulp, which is rich in lymphocytes, T and B zones, red pulp where the RBC macrophages and are there for the phagocytosis. There are these mucosa-associated lymphoid tissue that includes gut-associated lymphoid tissue, bronchus-associated lymphoid tissue, nasal-associated lymphoid tissue. So they protect the mucosal surface, gut, respiratory or urogenital tracts. So they are exposed to high antigenic load. Okay, because these are the openings of our body through which the antigen can enter very easily. So they are exposed to the high antigenic load. Okay, and they contains the lymphoid follicles with B and T cell areas, and the key structures are the Peyer's patches, tonsils, isolated lymphoid follicles. Okay, so yeah, so this also, okay, we can just go through. So um, in response to the B cell activation, they produces the antibodies. So these antibodies are called as immunoglobulins. They are Y-shaped. They produced by the B cells, specifically the plasma cells, in response to antigen, and their function is to recognize and bind specifically to antigen and help the immune system neutralize or destroy them. Okay, so if you will see the structure, it has chains. So it has two heavy chains. So in the Y shape you can say, so this is one monomer, this is another monomer. So this is a heavy chain. Okay, it is having two heavy chains, and there are two light chains. Okay, so long chains forming the inner part of the Y, shorter chains are forming the outer part of the Y. Okay, and both the chains are connected by the disulfide bonds, you can say here. And in the region, in these chains you have the region. So this part, this three bottom part, you can see these are the constant region. Okay, and the top part is the variable region. So variable region is at the tip of the Y, both heavy and the light chain, both heavy and light chain. So it binds specifically to the antigen. So each antibody has a unique variable region for a specific antigen. Okay, so that is why they are called variable, because they are different against different antigens. Okay, and the constant region, which is common in all the antibodies, so it determines the class of antibody basically, and it has the effector function. It interacts with immune cells, and these are called as with the help of Fc receptor and complement system. Okay, so the top portion is called the Fab region. This region is called the Fab region, fragment antigen binding. Okay, so this top part can bind to the antigen, and the bottom part is called the Fc region. So it is the fragment crystallizable. It is the stem of the Y, and it interacts with the immune cells and mediate immune response. Okay, so if any antigen is being coated by the antibody. So the top portion of the antibody binds to the antigen, and the bottom portion of the antibody will go and bind to the immune cells. Okay, so this bottom portion is called as Fc region. So the immune cell which can bind to the antibody, that has the Fc receptor, because this top region is binding to the antigen, so the immune cell should bind from this region. So the Fc receptor should have by the immune cell, so that it can bind to the antibody. Okay, so yeah, so this antibody is being released by the B cells. Then in the T cells you have these helper cells, cytotoxic cells, regulatory cells, and memory cells. Okay, so maybe we'll okay, so first thing is T helper cells. Okay, the marker is CD4 positive. All these cells having a specific marker which are called CD4, and that is why they are called as CD4 positive. So they help to activate the B cells, macrophages, cytotoxic T cells via cytokines. Okay, and then there is this cytotoxic T cells which is having the CD8 marker, and they are called CD8 positive. So they kill the virus infected cancer or foreign cells. Then there is regulatory T cells, Treg, CD4 positive, CD25 positive. So they suppress the overactive immune response and maintain the tolerance. Okay, so they help in tolerating the self-reactive response. So they help in suppressing the autoimmunity basically. Okay, then there is this memory T cells. These are having variable markers, so they provide faster response on re-exposure to the same antigen. Okay, so for T cells, if I'll talk, so there are, in order to activate, okay, it is having a 2 or 3 signal mechanism. So T cells, they cannot recognize the free antigens. Okay, so any antigen which is free in the surrounding, they cannot recognize them. Okay, for T cell to recognize any antigen, it has to be presented by an MHC molecule. Okay, MHC marker. Okay, so they need antigen presentation by antigen presenting cell. Okay, so the soluble antigen it cannot recognize. They want antigen to be presented by other cells. Those cells are called antigen presenting cells. Okay, that includes dendritic cells, macrophages, or the B cells. All these three are antigen presenting cells. They present antigen to the T lymphocytes with the help of MHC molecules. Okay, so the first one is the antigen recognition. Okay, so antigen recognition. So yeah, so this is, you can see this is a, let's see first this one. So this is a antigen presenting cell. It is a dendritic cell. It is an antigen presenting cell. Okay, here you can see on the top of on the surface of this antigen presenting cell, it is having MHC molecule. Okay, from the MHC is there. On top of the MHC, antigen is being loaded, and this antigen can be recognized by the T cells. Okay, and this for recognition, it has a receptor, so it is called as T cell receptor. So T cell receptor recognizes the antigen which is presented to it by MHC present on antigen presenting cell. Okay, so that is the first signal. So CD4 positive cells, they recognize the MHC class 2. Okay, so this is a T helper cell which is CD4 positive cell. They recognize the antigen which is being presented to them by um, um, MHC class 2. Okay, so this is MHC class 2. It is it is presenting an antigen, and T helper cell can recognize the antigen which is being presented by MHC class 2. CD8 positive cell, they recognize the antigen which is presented to them by MHC class 1. Okay, so how the difference? So difference here is in the form of antigen processing. So MHC class 2 presents the antigen which are extracellular antigen being processed and then presented, and then MHC class 1, they present the intracellular antigens, for example the virus. Okay. So here you can see, so this is the signal 1, T cell interacting with the antigen being presented by MHC molecule by antigen presenting cell. Then the second signal is co-stimulation. Okay, so there is this CD28 marker present on the T cell. Okay, it can bind to the B7. Okay, B7 in case of this one, um, the antigen presenting cell. Okay, so as it is already mentioned, the antigen presenting cells are dendritic cell, macrophages or B cell. So if it is a dendritic cell or macrophage, then the co-stimulatory ligand will be B7. Okay, here you can see, and the dendritic cell it is B7. If it is a B cell, then the co-stimulatory thing will be CD40 and CD40L ligand receptor. Okay, so that is what is the co-stimulation. So here B7 and C28, which is present on the T cell that interacts, that is a co-stimulation. So here you can see the CD28 is the receptor and ligand is B7 present on the macrophage. In case of the B cell interaction or antigen presentation by B cell, the CD40 ligand is present on the T helper cell and its receptor is there on the B cells. So that is basically a second signal work as co-stimulation for the activation of the B cell. So first signal will be antigen recognition, which is antigen presentation to the T cell by MHC, then the co-stimulation. So this ensures activation happens only during true infection, okay, which is not self-reactivity. Okay, without co-stimulation the T cell becomes anergic, which is nothing but being non-responsive. Okay, so that is the second signal. So these two signals are involved. First is the antigen recognition, then second is the co-stimulation, and then in some cases there are also a third signal, which is release of certain cytokines by the antigen presenting cell, which also give an additional signal, so that altogether activates the T cells. Okay, so in case of CD4 positive helper T cells, so this is the third signal, you can see, right? So when the third, after the all the recognition complex has been formed, co-stimulation is there, then the third signal, which is cytokine release by the antigen presenting cell, is happening. Under the cytokine release, the CD4 helper T cell, it gets differentiated. Okay, so based on which kind of cytokine signal it is getting, it will be differentiated. So that's why these cytokines play very important role in the immune system. So if the cytokine which is released is INF gamma or interleukins 2, then the cell CD4 positive T cells will be differentiated in Th1 subtype. So Th1 subtypes, what is the function? It activates the macrophages and cytotoxic T cells, which is cell mediated immunity. Okay, so and then if the Th2 is the subtype which is under the action of IL4, IL5, IL13, so it activates the B cell for antibody production. Okay, antibody production, which is humoral immunity. Okay, then Th17, under the influence of interleukin 17, it recruits the neutrophils, fights extracellular bacteria and fungi. If there are T regulatory cells under the release of IL10 and TGF beta, so its role is suppressing the immune response. It prevents the autoimmunity. So based on what kind of cytokine is being released, what kind of subtype of T regulatory cell will be formed, and what will be the function. Okay, so they are not directly killing the cells, but they are doing some downstream action which is helping to kill the foreign pathogen. Okay, for example, helping the B cell to produce antibody, recruiting neutrophils and suppressing the immune response, activating the macrophages, activating the cytotoxic T cells. So they are basically helping in killing them foreign pathogens. Okay, so those are the helper T cells. Then there are CD8 positive cytotoxic T cells. So after activation, they recognize and kill the infected or abnormal cells, displaying the foreign peptide on MHC1. Okay, so mechanism of killing. So there also you can see. So this is a cell infected, okay, which is having intracellular pathogens. Okay, so here you can see. So this is a target cell. It is presenting the pathogen on MHC1, which is being recognized by the T cell receptor of CD8 positive T cell. Then there is a second interaction also, which is Fas and Fas ligand interaction. Okay, and due to that activation happens. Okay, you can see the T cell receptor MHC class 1 molecule presenting the antigen extracted from the pathogen. It is activating the cytotoxic T lymphocytes, and then it is releasing the perforin and granzymes, which kills this cell directly. Okay, so this particular cell only having the infection, it is an intracellular pathogen. Okay, for example virus or the cancerous cells. So when the interaction takes place, this T cells gets activated, releases the perforin and granzymes, and uh, kills that particular cell, which is becomes an apoptotic body. So it performs the apoptosis. So the T cell releases the perforin, it forms pores in the target cell membrane, and the granzyme, it enters and triggers the apoptosis, which is programmed cell death. So both together perform the function. So the Fas and Fas ligand interaction. So this TC cells, they express a Fas ligands which can bind to the Fas receptor on target, that also induces apoptosis. Then there is a cytokine mediated killing. Okay, so the secretion of IFN gamma and TNF alpha, they also inhibit the viral replication and enhance the macrophage activity. Okay, so these are the different methods, a mechanism of killing the antigen presenting cell to the CD8 positive T cells, and either it can kill by perforin granzyme pathway, or Fas-Fasal interaction, or cytokine mediated killing. Altogether it will either let the cell die by the programmed cell death mechanism, or it if it is a viral infected cell, it will try to inhibit the viral replication and enhance the macrophage activity. Okay, so and then there is also the regulation of this T cell response. So these T regulatory cells, they release IL-10 and TGF beta, so they suppress the T cell proliferation and inflammatory cytokines. So the checkpoint molecules, these two act as brakes on T cell activation and maintains the tolerance and prevent autoimmunity. And there are also these memory T cells. Okay, effector, as the most effector T cell dies, few memory T cells remains in the central memory and the effector memory, and on re-exposure they perform the immediate strong secondary immune response. Okay, so in short, it is the antigen presentation or MHC and TCR binding. Then second one is the co-stimulation of CD28B7 activation, then differentiation into from CD4 positive to Th1, Th2, Th7, T regulatory. They release the cytokine and perform the immune regulation. CD8 positive cytotoxic T cell, they kills the infected cell via perforin granzyme or Fas ligand interaction, and they also form the memory T cells for the future responses. Okay, now if I'll talk about the B cell activation. Okay, so B cell activation, it is either T cell dependent or T cell independent. Okay, so T cell dependent, that means it requires the T cell help. Okay, and the T cell independent one, which does not require the T cell help. The antigen type for T cell dependent, it is protein antigens. For T cell independent, it is non-protein antigen, for example polysaccharides or lipids. And for example this one, it is either viral proteins, bacterial toxins, and for this non-proteins ones, the antigen could be bacterial capsules, for example in pneumococcus. Okay, just a minute. Okay, so in the T cell dependent, which is the main pathway for the B cell activation, so this is the most common and strongest mechanism which gives rise to high affinity antibodies and memory cells. Okay, so there are several steps. So first step, as in case of T cell, here also it is antigen recognition. Okay, so the B cell receptor binds a specific antigen. As the T cell having a T cell receptor, the B cell is also having a B cell receptor. Okay, So the B cell receptor, it binds a specific antigen. What is a B cell receptor? So B cell receptor is nothing but the integrated antibody molecule present on the B cell surface. So here you can see this is an antibody which is embedded on the T cell surface. So antibodies are of two types, one which is present on the surface of the B cell and another one which is present in the plasma, which is the soluble form. Okay, so uh, just a minute. Okay, so yeah, so the first one is the B cell receptor binds a specific antigen. So the antigen, so once the antigen is bound to the B cell, it is now internalized or endocytosed. Okay. Hmm. Just a minute, please. Yeah, so for the T cell dependent B activation, there are these steps. So first step is antigen receptor. So B cell receptor, which is nothing but the antibody molecule, it binds to the specific antigen. So antigen is internalized. Okay, once the antigen is bound to the antibody, it is internalized into the B cell. So inside the B cell, the antigen is being processed and fragments are displayed on the MHC class 2 molecule on its surface. Okay, so this is the MHC molecule. It presents the differentiated or internalized and processed antigens. Sorry. Then the interaction with the helper T cell will begin. Okay, so the helper T cell, it can bind to the antigen which is being presented to them by the antigen presenting cell, which here in this case is the B cell. Okay, and B cell is also presenting with the MHC2 molecule. Okay, so B cell, which is presenting the antigen on the MHC molecule, it binds to the T cell receptor on the helper T cell. Okay, so the CD4 positive helper T cell, which is already activated by the same antigen via a dendritic cell, binds to the B cell. Okay, again with this antigen is already being presented to the helper T cell, it is already activated. Okay, so the TCR of the T cell recognizes the antigen MHC2 complex on the B cell, and the CD40, which is present on the B cell, binds to the CD40L, which is present on the T cell, which is a co-stimulatory signal, the second signal, and this two-way interaction is essential for the B cell activation. Okay, now this interaction is essential for the uh, B cell activation, because the receptor which is there on the CD40. So when this second interaction happens, the B cell gets activated. Okay, so in the other case scenario, you can see the receptor was, the co-stimulatory receptor was there on the CD20, this helper T cells. Okay, the CD28, which is the co-stimulatory receptor. So here the T cell was being activated, and this is having the receptor for the co-stimulatory signal. And here in case of, for the B cell activation, you can see the receptor is present on the B cell, and then the B cell gets activated. Okay, I hope this is clear. So then the cytokine help is needed. Okay, so here you can see B cell receptor bind to the antigen, internalizes, then processed, presents with the help of MHC, binds with the T cell receptor, then the co-stimulatory signal and of this CD40 ligand, and then there is a cytokine release. Okay, so the helper T cell, they secretes the cytokines, so these IL4, IL5, IL6, IL21, different kinds of interleukins. So interleukins were interleukins, the communicatory molecules between the lymphocytes. Okay, those are called as interleukins. These are cytokines. So these cytokines promote B cell proliferation. Now the B cell is activated. Okay, then the T cell is releasing some cytokines, and these cytokines, so depends what kind of cytokine is being released. Okay, so they allow or activate the B cell for the B cell proliferation, that means the clonal expansion, the class switching. So the B cell receptor which is present on the this surface, it is IgM. So when it gets activated, it start producing different classes of antibody, IgG, IgA, IgE, for different kinds of action. Class switching, then the affinity maturation. Okay, so more affinity for the antigen by the B cell. Differentiation into plasma cells and memory cells. Okay, so this plasma, when it differentiated into the plasma cells, so it is a cell which secretes more and more antibodies. The memory cells, which are present for the long term and help in the when the second exposure is there, and generate more and differentiated more, activates, gets activated faster, and then again starts secreting the antibody again that specific antigen. Okay, so this is how this entire process takes place. Okay, so the outcome of the activation will be clonal expansion. So clonal expansion will be activated B cell divide to produce identical clones recognizing the same antigen. Okay, the heightened response. Then the plasma cell formation. So the effector cell, these are the effector cell which secrete the large amount of antibodies. So B cells are undergoing first clonal expansion, more and more B cell that are specific against that particular antigen, and those again gets differentiated into the plasma cell, and those plasma cell are the effector cell which will produce more and more antibodies against that specific antigen. Okay, then the memory cell is also formed, which are the long lived cells providing faster immune response upon re-exposure. Class switching happens, so antibody type changes without altering antigen specificity. Okay, and affinity maturation, so somatic hypermutation in variable region, so higher affinity antibodies are being produced. Okay, so the antibody which is binding to the antigen, so that top of the antibody was the variable region, right? So that variable region, there are certain more mutations are accumulated, so those are called somatic hypermutation, high, because the faster proliferation is happening, because of the faster replication there are errors, because of the error there are mutations. Those are called somatic hypermutation, very high rate of mutation only in the variable region. Okay, so and if due to that mutations the maturation or the affinity of the antibody is becoming more towards that particular specific antigen, then it is called the affinity maturation. Okay, so this is the outcome of the activation of the B cells. Okay, then the next one, which was the T cell independent B cell activation. So there are some antigens which can be activate the B cell without T cell help. So these are the non-protein antigens. Okay, and the mechanism will be repetitive epitopes. Okay, cross-link many these BCRs on the same B cell surface, and then they give the activation signals. Okay, and there are rapid but weak response. They produce mostly the IgM antibodies, and it has no memory or class switching. Okay, so this is the T cell dependent pathway, this is the T cell independent pathway. In the dependent one, you can see there is antigen which is first interacting with the B cell receptor, which is nothing but the antibody molecule. Okay, and then there are all these co-stimulatory signals, cytokine release, which is happening, which is activating the B cell. Upon activation, it is undergoing clonal expansion and differentiation into plasma cells and memory cells. Plasma cells are making more and more antibodies, there are class switching, there is affinity maturation is happening. Okay, then this is the T cell independent pathway, which is the pathogen being recognized by the B cell receptor, which is non-protein in nature, and this is also making the plasma cells but no memory, and mainly producing the IgM antibodies. Okay, so yeah, so the feature will be so this is the T dependent, T independent, we have already discussed. Functions also, function of the activated B cell, antibody secretion. Antibody, what do? They neutralize the toxins and microbes. The opsonization, which is nothing but coating the microbes for phagocytosis. Complement activation. Okay, so these antibodies or activated B cells, they trigger the complement cascade for the pathogen lysis. Antigen presentation, they present the antigen to the T cell via MHC2. There is a memory generation. So overview will be antigen binding first, then internalization, then MHC2 presentation to the T helper cell contact, CD40 ligand plus cytokine release, which activates the B cell with that will promote the class switching and affinity maturation, and also the plasma cell and memory cell differentiation. Okay, so there are different types of antibodies. Okay, and these are divided into different class based on their different structure in their constant chain basically. Okay, here you can see the constant region is different in different of these antibody classes. So these are IgM, IgD, IgG, IgA, IgE. Depends on which particular molecule constant region is present. If the mu chain is present, it is IgM. If gamma chain is present, gamma chain is present, IgG. Delta is present, it is D. Epsilon, E, and alpha is A. Okay, so yeah, so there is IgG, gamma chain and all I have already told. So how they are structures, there are different structures. IgG remains as monomer. It is present in blood and placenta, and it provides long-term immunity. Okay, present in blood and placenta. Okay, this is the only antibody which can cross this placenta barrier, cross through placenta, sorry. So IgA, it presents in a dimer form. It is present in the secretions and the mucosa, helps in the mucosal defense. IgM, it is a pentamer. It is present in the blood and lymph. It is basically the first response and the agglutation. IgE is a monomer. IgD is also monomer. E is bound to the mast cell, and allergy and parasite defense is the function. IgD, it is present on the B cell surface as a B cell receptor, and it helps in the B cell activation maybe, and it has a very unclear role because IgM and D both are present on the B cell surface as a receptor. And then there are these two terms, which is affinity versus avidity in case of antibodies. So affinity is how strongly it is binding to the antigen. Okay, so it is the strength of a single antibody antigen interaction. Each IgG antigen binding site typically has high affinity for its target. Avidity refers to the strength of all interaction combined. Okay, so all together, so to an antigen, how many antibodies are being able to bind? So IgM has low affinity but antigen binding sites, low affinity antigen binding sites, but there are 10 of them. So avidity is high. So that is how many antibodies can bind to the antigen, and how specifically it binds to, how strongly it binds to the antigen. So those are two different things being referred by two different terms, affinity versus avidity. Okay, so yeah, this is the constant chain, variable chain, both. So this is the variable chain of the heavy region, heavy chain, light chain. Okay, so here you can see the variability is, so if this is the heavy chain variable region, so this entire peptide. So there are these regions in the light chain also. There are this region which are having high variability. Okay, so they are also referred as heavy chain variable region 1, 2, 3, HV1, HV2. Okay, and sorry, no, it is highly variable region 1, 2, 3. Same goes here. Okay, and this is, suppose in the light B chain it is denoted. So in the entire light chain variable region only, in that entire peptide portion, there are certain portions where the variability is very high. Okay, so when you can see the protein is now in the native conformation they are seeing. Okay, there are three reasons. So these are called as CDR1, 2, 3. Okay, and when in the, so these are presented with the red color in the antibody structure when you are seeing. So these are the red color of the CDR1, 2, 3, and there you can see all these three are lying in the top notch where it binds to the antigen. Okay, so as each antibody has different affinity for different antigen, that is because it has so much high variability. Because of variability, it can bind to different kinds of antigen. Okay, so this is the reason on top of here, which is highly variable, and due to high variability it can bind to different kinds of antigen, and it is the one which is making the antigen binding site. Okay, So the structure wise it is similar to the T cell receptor, you can see here also. And the TCRs, it is having distinct subset of T cell. They bear an alternative receptor, either alpha beta receptor, gamma delta receptor. There are different theories, germline theories, for so much variability. Okay, so either there are separate genes for each different antibody chain, and antibody repertoire is largely inherited, but and there is also somatic diversification theory. So a limited number of inherited B region sequence undergo alteration within B cell during the lifetime of an individual to generate the observed repertoire. Okay, so if you compare the number of genes and then the type of how many variable regions are there, it is too high. So the germline theory does not correlate. It is possible that through so much diversification happens during the B cell development, and that is why so much variability is arising in this variable region. Okay, so here it is given, if this CDR123, if it is original genomic segment, you will see you have this kind of structure where it is the all the exons. This is exons with the introns present in between. Okay, here. So this is called a V segment, this is called a J segment. Okay, okay, so this is VJ, and this is the constant region, okay, of the entire. So this is the germline DNA. Then in the light chain and the heavy chain. Okay, so in the heavy chain, first this D which is responsible for diversity. So D region, which is present only in the heavy chain, so D region combines with the J region. Okay, so somatic recombination. Okay, it is basically a recombination event. So DJ recombination, and then in case of this one, there is after DJ, then there is for this one VDJ recombination, for it is VJ recombination. Okay, then there is a primary transcript of RNA is formed, then which undergoes splicing, which makes the mRNA, which undergo translation. And here you can see after the translation, the protein is arranged, or the conformation of the protein is arranged in such a way that this J domain and the V domain is coming here on top of this, where the antigen binding site is present. Okay, here you can see in the variable region, this is the variable region of the heavy chain. Okay, here you can see in the antigen binding site, this DJ region is coming. Okay, so this is the recombination events. So here you can see multiple contiguous V chain segments are present at each immunoglobulin locus. Okay, so for this is for the, so there are delta and kappa light chains. So here you can see there is this kind of arrangement, variable V uh, V gamma kappa 1 V kappa 1, and then there is kappa 2 3 4, and there are also 1 2 3, and there are 30 such variable regions are present. Then there is intron, then there is a J, which is J 1, then there is a constant 1, then J2 which is constant 2, like that. And here there are several J and then C. Okay, so there are several recombination can happen. Okay, so if the light chain contains this type or kappa type, whichever of these it will contain, so it will first VJ recombination. Okay, and here in this one, and there also the VJ recombination, and then it binds to the constant kappa chain, and here also the constant one also are different in different types. Okay, so that also being together. So this is basically a probability, or you can say how many different combinations can be generated. Okay, so if there are total 38 kappa chains and 5 joining chains, so 38 into 5, that many combinations can be made, and there is one constant. And here in the constant change also there are 9 types, so altogether multiply, and that many combinations can be arise. Here also D regions are also, there are different type of segments. Okay, so whichever is being chosen, only one of them is being recombined to the J, one of the J, and then that 1D 1J, and then that will combine with one of the variables of the heavy chain. So there are different kind of combinations. Suppose D1 is combining with J1 and it is combining with V1, so that is one combination. If D1 is combining with J2 and then that is combining with V1, that is one combination. Again the V1 combining with J3 and it is combining with V1, that is another combination. So there are so many combinations can be generated, okay, and that is how the variability is arising here. Okay, so there is also this rule. Okay, so the gene segment flanked by an RSS with 12 base pair spacer typically can be joined only to a one flanked by 23 base pair spacer, and this is known by 1223 rule, which is discussed here, which I have already discussed also. I'll not go in the details, but that is a basic a thumb rule for this particular topic. Okay, for the joining of this VDJ segments, 1223 rule. And this is the rearrangement. Gene segment is guided by flanking DNA sequences. Okay, so all these things I think we have discussed individually. Today we are just summarizing, so I'll not go in the details. Okay, yeah, so this is the somatic hypermutation, you can see. So after the primary response, secondary response, you can see in the CDR3 region more and more mutations are being generated, and that is helping in increasing antibody affinity. So those are selected which are having gathered those mutations specifically in the CDR3 regions, which is the variable region, which is present in the antigen binding site, which is increasing the affinity of the antibody against that particular antigen. Okay, this is also the, yeah, for the T cell receptor generation also, the, as I've shown, the antibody structure and the T cell receptor structure is the kind of the same, so it is also the same kind of rearrangement is being happening. Okay, so here you can see there are these many heavy chains, and then kappa and this gamma chain, and how many D segments are there, how many J segments are there, and based on that how many number of B gene pairs can be generated, number of junctional diversity, total number of diversity, how many it could be. So these many diverse or variable regions can be generated. Based on different combination it can be rearranged. Okay, so this is again, yeah, so for the MHC1, so MHC molecule which present antigen to the T cells. Okay, so there is also an MHC restriction. So antigen presentation happens when the and this is also specific. So for example, here you can see the recognition is happening. Here the antigen is being presented by MHC and it is being also fitting perfectly and recognized by the T cell. Okay, that specific T cell. Here you can see it is fitting perfectly with the MHCB, the MHC molecule, but it is not fitting perfectly with the TCR, so there is no recognition. Similarly, here there is a different antigen. Okay, the T cell and MHC are same, but the antigen is different, and due to that it is not able to bind to the T cell, and there also no recognition is happening. So the MHC polymorphism is there, which affects the antigen recognition by T cells by influencing both peptide binding and the contact between the T cell receptor and the MHC molecule. Okay, and yeah, so this is how the antigens are being prepared for the MHC presentation. So cytosolic pathogens, so they are degraded in the cytosol. There are the peptide which can bind to the MHC class 1 and they are presented to the CD8 positive T cell, and the effect will be the cell death. Okay, if there are intravesicular pathogens, so they are endocytic vesicles presented by MHC class 2 presented to CD4 positive, and effect will be the activation to kill intracellular bacteria and parasite. If from the B cell, so endocytic vesicle again presented by class 2 to CD4 positive, and the activation of B cell secretion of immunoglobulin to eliminate the extracellular bacteria or toxins. That is the process. So this is also cross presentation of exogenous antigen by MHC class 1 molecule by dendritic cells. See, internalization. Okay, so this is a kind of example of cross presentation which is also done by the dendritic cells. So MHC1 presents the intracellular pathogen. So this is one example of cross presentation. Here also internalization is happening. Phagolysosome is degrading and making the antigen which is being loaded on the MHC molecule and being presented by the MHC class 1. And this is the presentation of cellular antigen by MHC class 2 molecule. Okay, so this is the autophagosome which is cleaving the self antigen, and so this is, so initially when the MHC is ready, it is bound to some other molecule which is, or antigen, which is called as clip. So when these two combine, it replaces the clip and then being loaded on the antigen as MHC, and then MHC goes and being presented in the cell surface. Okay, this is the virus infected cells. Okay, so the T cells which are presenting the intracellular pathogens. So this is the virus which is intracellular pathogen now. So here the viral genomic DNA gets transcribed, translated, and then goes to the ER, and in the ER only it interacts with the this MHC, get loaded on the MHC, and then goes and being presented by the MHC class 1 molecule on the surface. And these are the proteasomal machinery, which are the peptides are generated by ubiquitinated proteinase in the cytosol by this proteasomal degradation. Okay, so here you can see this is the MHC class 1, calnexin is bound first, then calreticulum, all these molecules are present. Okay, so the viral RNA is making the proteins and everything. So yeah, so this is the one also we have discussed in the class. So the same thing, I'll not discuss in the detail. So this is initially how the invariant chain directs newly synthesized MHC class 2 molecule to acidified intracellular vesicle. Okay, so initially it is loaded with this particular molecule which is called as clip. Okay, and then the clip is being replaced by the internalized pathogen, and then internalized pathogen is being presented on the surface. So there are several genes which are involved in help. For example, this HLA, it also helps. So if there is a mutation in the HLA gene, then there is a difficulty in the antigen presentation, and if the antigen is not being able to being presented to the effective or immune cells, then there is a shutdown of the immune response. Okay, so this is also via the ubiquitinations, the peptides are being ubiquitinated, loaded on the MHC molecules. Okay, these are the techniques for detection, immunology used in the immunological studies. For example, the antigen antibody interaction are being taken or associated or work as an effective mechanism in the laboratory. For example, visualizing the protein bands, identifying the protein bands, okay, by Western blot and by ELISA. So this is about the B cell maturation, how the B cell is getting matured. Okay, B cell activation. So this is the after the maturation, it goes to the lymph nodes, and how it gets developed, it interacts with the pathogen and then goes to the germinal centers. Here they are called as centroblast in the dark zones of the, and then after interaction it goes to the light zone where it called as centrocytes. From the centroblast, it undergoes somatic hypermutation and the affinity maturation. So these are the effects. Okay, so this, as you can see, in the activated B cells, somatic hypermutation is happening. After that, it is going to the dark zone where it is going under the high affinity maturation. High affinity one are being selected, then the class switching is happening, then the interleukins release, cytokine release is happening, making more and more plasma cells, differentiation into the plasma cells. Okay, and then this is the complement system that also complements the immune cell response. Okay, so alone the antibody production or the T cell activation will not help. So these are the complement system which tries to complement or complete the immune response. So these are the complement proteins. So they get uh, fragmented, smaller portion, larger portion, and then coats. And there are three uh, functions. They help in lysis of the cells. The infected cell they can lyse, they can make this membrane attack complex and help disbalance the osmolarity, so that it will crush the cell, and then it will burst the bacterial cell or any pathogenic cell. Then there is opsonization, that means it coats, the complement proteins coats the pathogen, coating by the antibody, and then it is recognized by the macrophages or antipresenting cell, and then the internalization, phagocytosis will take place. And also these complementary proteins help in enhancing the inflammation or the immune response, releasing the histamines and granzymes. Okay, so this is the function. This is the entire pathway, how the complement proteins are associated, formed, activated one by one, that we have talked through. Okay, I'll not go through in detail. So this is the entire process. This is the cytokines. Okay, so what cytokine has what effect? IL-2, IL-4, IL-5 have help in the B cell maturation. If IL-4-5 release, it helps in B cell class switching. If IFN gamma is there, it inhibit IL-4 and then inhibits the class switching. So these are different kinds of cytokines. Okay. Yeah, so if there are immune dysfunction, if there is hypersensitivity, it will cause the allergy. If there is autoimmunity, it becomes self-reactive, then also it causes autoimmune diseases. If there is immunodeficiency, then these compromised immune diseases are generated. Then also if the immune dysfunction is there, transplant rejection will be there due to MHC mismatch. Okay, this is the summary for hypersensitivity. Okay, so I'll just quickly go through the questions. Okay, so I'll just go through. I have also put the correct answers here. So the which of the following is the primary lymphoid organ? Okay. So as primary lymphoid organs, there are two, bone marrow and thymus. Thymus is listed here. Correct answer will be thymus. Which are the responsible for antibody production? Which cells? So B cells, which get differentiated into the plasma cells, they produce the antibody. So correct option will be the plasma cells. Which immunoglobulin is the first to appear during primary immune response? Answer will be IgM. It will be the produced first and later followed by IgG during the class switching. Which antibody class can cross the placenta? If you remember, IgG is the only antibody that crosses the placenta and provides passive immunity to the fetus. Which cell types acts as a bridge between innate and adaptive immunity? I am saying from the very beginning, dendritic cells, as they present antigens to the T cell and links the innate and adaptive immunity. MHC class 1 molecule, they present peptides to which one? So they present peptide to the CD8 positive cells. MHC class 2 molecule present peptide to CD4 positive cells. The complement system can be activated by all. So yeah, so there are, for the complement activation, there are three pathways: classical pathway, alternative pathway, lectin pathway. So if I'll just show you. Okay, so this is the classical pathway, where one set of protein starts with the C1QR2S2. Then there are, if the lectins are involved, there is another lectin pathway, and in between that there is an alternate pathway that goes on, that is called as alternative pathway. So only because of these three pathways the complement system will be activated. So cytokine pathway is nothing pathway for activation of the complementation pathway. The antigen binding site of an antibody is formed by the variable region of the heavy and the light chain. Okay, FC portion is the bottom one, which binds to the FC receptor. Constant region of the heavy chains, no. Hinge region is also not involved. It is lies in the variable region of both heavy and the light chain. Which of the following is not a feature of innate immunity? Non-specific, correct. Innate immunity are not specific. Memory formation, no. For innate immunity, there is no memory. It is only for the adaptive one. And rapid response, correct. Physical barrier, that is also correct. It includes the skin and all the mucosal areas. Okay. So which cytokine is mainly responsible for the fever? So the interleukin 1 along with IL-6 TNF alpha, it acts as an endogenous pyrogen. So which type of hypersensitivity is mediated by IgE? So the answer is type 1. It is allergy anaphylaxis is mediated by IgE, mast cell granulation. So in the central tolerance, so there are this self tolerance pathway which I have discussed in the last class, I guess. So there I have talked about the central tolerance and the peripheral tolerance. So central tolerance, when they in the primary lymphoid organ, so central tolerance is a word where the immune system is prepared to not interact very efficiently with the self cells. Okay, so a control, a checkpoint. So if so first the checkpoint is there in the primary lymphoid organs, that it should not react with the self cells or the self protein. It should not show a high response, and that is the central tolerance. And if it passes the central tolerance, goes to the secondary lymphoid organ, there is another tolerance which is called the peripheral tolerance, and there is also a check for the these T lymphocytes or the B lymphocytes, and that is with the help of T regulatory cells. Okay, so that is the central tolerance. So it is there in the thymus, as the thymus is the and it is for the self-reactive T cell, then the answer will be thymus. Both thymus and bone marrow are the locations for the central tolerance. If it is asking for the T cell matures only in the thymus, the correct answer will be the thymus. So it central tolerance eliminates the self-reactive T cell in the thymus, and that is a process called negative selection. Positive selection was when it is recognizing self-MHC. That is positive selection. Check, that is taken. If that taken cell is reacting very efficiently with the self antigen, then that is eliminated. That is negative selection. That is part of central tolerance. Then the antibody class most abundant in the mucosal secretion, that is IgA. Okay, it is secretory IgA. It protects the mucosal surface, respiratory, intestinal, urogenital tracts. So the process by which neutrophils move towards site of infection is called as chemotaxis. It is directed movement of immune cell towards chemical signal at the infection site. The Fc region of antibody is responsible for. Okay, so it is responsible for antigen binding. No, it binds by Fab region. Okay, so it is responsible for class determination and effector function. So Fc region bind to the Fc receptor and complement it determine the antibody's effector. Which of the following is best defines opsonization? So opsonization is nothing but the coating of pathogen for enhanced phagocytosis in the pathway of the complement activation. So which of the following statement about the complement system is true? It is part of the adaptive immunity. It is enhances phagocytosis and lyse pathogen. Correct. It acts only against virus. No. It is produced by T cell. No. So it enhances the phagocytosis and lyse pathogen. So that is the correct option. So this is it. This is a summary for whatever we have gone through in the last weeks. Okay. If you want to ask anything, any doubt, you can ask me now. Anything, any part you didn't understand? Any doubt? If there is no doubt, then I'll conclude the session. I hope it was clear. If you want to ask anything, there is one more class remaining. You guys interact, ask me the question. You can stop me in between. Okay. So if no question for today, I'll conclude the session for today. And thank you for joining. We'll see you in the class next coming Tuesday. Okay.